So, good morning, everyone. So, we'll be talking about hemoglobin structure and function this morning, and it is necessary for you to have a good understanding of hemoglobin structure and function in order to appreciate uh, physiology and pathology of sickle cell anemia. Okay, so hemoglobin is found in red blood cells. As you are aware, you have three major types of cell. Okay, these cells are white blood cells, red blood cells, and platelets. So we'll be talking on red blood cell this morning, and the content of red blood cell is hemoglobin. Hemoglobin is the content of red blood cell. Matured red blood cell has no nucleus. Matured red blood cell has no nucleus. Okay, so hemoglobin constitutes like 95% of the content of the red cell. So hemoglobin, like I said, has each cell has like 640 million hemoglobin molecules. Each red cell has about 640 million hemoglobin molecules. Okay, so and each hemoglobin carries 4 molecules of oxygen because each hemoglobin has 4 heme group, and each heme carries a molecule of oxygen. Okay, so if you multiply 4 molecules of oxygen by 640 million hemoglobin, so each red cell will be carrying 4 times 640, which is like 2.56 billion molecules of oxygen. So this is the structure of hemoglobin. Hemoglobin consists of, like I said, it has heme and globin, and that is why it is called hemoglobin. Okay, the globin is polypeptide, and there are 4 polytypes, 4 polypeptide per hemoglobin and 4 heme. So this lecture we're talking about heme, and we'll be talking about globin. We'll be talking about their synthesis, we'll be talking about their breakdown. Then we'll talk about the function of hemoglobin. We'll talk about abnormal hemoglobin, because when you have abnormal hemoglobin, that's when you have sickle cell anemia, and this lecture is important for you to appreciate the structure of hemoglobin. You need to know the structure, the normal structure, before you start talking about the abnormal. Okay, so another abnormal type of hemoglobin is thalassemia. So what's the difference between thalassemia and sickle cell? One has a quantitative defect and one has qualitative or structural defect. Sickle cell has a structural or qualitative defect. Thalassemia has a quantitative defect. So these are the abnormal type of hemoglobin. There are other types of abnormal hemoglobin. For example, if the iron that is the heme has a iron that is responsible for carrying the oxygen, and it has to be in ferrous state. So if it is in ferric, then it has what is called oxygen methemoglobin. So if it is in ferric state, that's Fe3, Ordinarily it should be Fe2. So if it's in Fe3, it's also an abnormal form of hemoglobin. You could also have sulfur hemoglobin if there is sulfur molecule in the hemoglobin. It also is another type of abnormal hemoglobin. So this is another structure of hemoglobin. You can see that there are 4 polypeptides, one pair that looks alike and the other pair that looks alike. And each polypeptide, which is the globin, folds around a heme molecule. So, and don't forget that heme is responsible for carrying the oxygen. Okay? Heme carry, so you can see that there are 4 hemes in this diagram, and this is it. This is the first heme. This is another heme. This is another heme. So this is the structure of hemoglobin. So the polypeptides are labeled as alpha, beta, delta, gamma, zeta, epsilon, theta. I hope you still remember all the Greek name of all the, alpha. So for adult hemoglobin, if you are not thalassaemic, you should have normal quantity of amino acid in your alpha and the beta chain. You can see the color, 2 chains have the same color, and the upper part also have. You can see that you have alpha 2, 2 alpha chains, beta 2. And how many heme do we have in us? We have 4. So this is just one molecule of hemoglobin carrying 4 molecules of oxygen. And like I've said, you have close to 640 molecules in every red cell. So if you now multiply that by the number of red cells you have, you can then be able to imagine how many molecules of hemoglobin. So if you don't have adequate number of red cells, so what is that called? Anemia. So the number of oxygen molecule you'll be able to carry will be more. Is that correct? If you don't have adequate, it will be less. It will be less now. That's why you have shortage of blood. Then you have anemia, and you could be breathless. So all the functions that the oxygen does, you will not be able to do it. Okay? So like I said, you have 2 major types of hemoglobin. You have what is called alpha and zeta, which has the structure, that's the how zeta is written. Alpha and alpha like, beta and beta like. There are 2 major groups. You have mentioned alpha, beta, zeta. So it's either alpha and alpha like. The alpha like is the zeta. And why is it called alpha like? Because it carries the same number of amino acids. You can see it's 141 amino acids. Like I've said, globins are polypeptides. So they carry amino acids. Okay? But and also located on chromosome 16. Okay? But the beta, you can see the beta, the gamma, the delta, the epsilon contain 146 amino acids. So there are 2 major groups, and they are located on chromosome 11. Okay? So all the hemoglobins are synthesized before the red cell becomes mature. And the reason is that mature red cell do not have mitochondria. And then you need mitochondria for production of heme. It starts in the mitochondria, and the last 3 parts of it ends in the mitochondria. So hemoglobin is formed during development of the red cell in the marrow. So once it becomes mature, it is pushed into the peripheral. So if I collect your blood, I'm collecting mature red cell, and it lacks mitochondria, it lacks nucleus. So So if for you to have hemoglobin, the formation must have been complete before it becomes mature. So that is why I said 65% of hemoglobin is synthesized in the erythroblast. Reticulocyte is the last stage. There's still mitochondria. But once it becomes mature, it loses its mitochondria and also loses its nucleus. So don't forget that beta globin gene, that's chromosome, is carried on chromosome 11, and alpha globin gene is carried on. So how many amino acids you have in the beta globin gene? 146, and then 141. If you count 1, 2, 3, 4, 5, 6 like that, it will be 146 amino acids. So most of the mutation affects the beta chain, like in sickle cell. And that is why it is at beta chain number 6. Number 6. That's where the mutation takes place. Number 6, in which you have valine replacing glutamic acid. Okay? If you have valine replacing, what kind of amino acid is glutamic acid? Positively charged, negatively charged, neutral? Negatively charged. So what about valine? Is neutral. So there will be deficiency of charges. One that is neutral is replacing negatively charged, so it's going to affect the physicochemical properties of that hemoglobin, and that is what causes sickling of the red cell. Don't forget hemoglobin is inside, and all the 640 million hemoglobin at beta chain number 6, they all carry valine instead of glutamic acid. So there is bound to be changes in the structure because of the charges, of the difference in charges, and that is why you have sickle. I hope we are getting the rudiments why you have sickle cell. So what happens, you now start having sickling of the red cell because of this change in valine at position 6. So this red cell will now, what's the normal shape of red cell? Biconcave disc. God has made it in form of biconcave disc, so that they don't stack on each other. But you now have red cell that is sickled because of the change. So they will now, and we are talking about billions and billions and billions of red cells. So they will now, lie on each other and then occlude microvasculature. You have tiny vessels in the eyes, in the ears, in the penis, everywhere, in the bone. And when they occlude, what's the implication of that? They occlude the vessels, the arterial circulation. What is the implication? It will compromise oxygen, because oxygen, number one, it won't be able to carry oxygen to the end tissue. And when you don't have oxygen, what happens? No, it's not death like that. It's when you have ischemia, pain. What happens during anaerobic glycolysis is you don't have oxygen, the cell will make use of anaerobic glycolysis. And what's the end product of anaerobic glycolysis? Lactic acid. So you have lactic acid. So if you have plenty of acid in your body, what's the implication? Acidosis. Patient will become acidotic. And the implication of that is that we need to talk about it. It will now shift the hemoglobin dissociation curve to the right, so that the body is like a compensation. The body will be able to release the oxygen, the little oxygen it has. But the acidosis will cause pain, and then the occlusion because of lack of oxygen. Well, that's the pathogenesis of sickle cell, but this is the baseline, okay? So for you and I that are not sickle cell patients, I assume you are not sickle cell patients. So this is what you have. So you could have a combination of alpha and a non-alpha. I said you have alpha. and alpha like, which is zeta. Okay, you have beta and beta like, which is gamma, delta, epsilon. So in any combination, it has to be alpha or alpha like and beta or beta like. I hope that is clear. In any combination of hemoglobin, you cannot have alpha and and you cannot have alpha and zeta because they are the same type. It has to be alpha like or alpha or beta or beta like. You understand combination? Look and see this combination. You can see for hemoglobin A is alpha 2 and beta 2. For hemoglobin A2, A is also called A1. It's also called A1. So if you are not a sickle cell patient, these are what you are going to have. Have you heard about high performance liquid chromatography HPLC, that you can quantitate the different type of hemoglobin? Have you heard about that? Oh, that's what we do now for our sickle cell patients. We don't do hemoglobin electrophoresis, so-called genotype. We don't do that again because it causes confusion. Some people will look at it and say it's AS. Another person will say it's SS because it's very subjective. You understand? That cause a lot of problem. Many people are married today and they have SS because of a wrong genotype they got while cutting. Okay, but if you use HPLC, which we have here, high performance liquid chromatography, you will quantitate the amount of hemoglobin in the blood. So if your result is this, you can be labeled as AA. Is that not correct? But if you have S gene, the machine will quantitate the S too. Okay, if you quantitate the S, it will quantitate the C if you have C gene. So if your result, if the machine is saying your hemoglobin A is 15%, 10%, and your hemoglobin S is 80%, what is the genotype? AS will be 50-50 really, but most likely you won't get that result. Most of your SS, the machine will quantitate hemoglobin S as 80-90%. And A, you still have a little bit of A2 in, and F. Like everybody will have little quantity. Like even if you are AA, if I check the quantity of A2, it will be between 1.5 to 3.2%. And we still have hemoglobin F. Where will F be highest? Which group of patients or group of people? In babies. Why should it be highest? So if a newborn baby, if I check the hemoglobin now, then I'm going to be getting hemoglobin F of 80%. You can see it, newborns. Why is it like that? Because F is specially adapted to operate in low oxygen tension. In low oxygen tension, S hemoglobin F is specially adapted to operating. So do you have very high oxygen tension in utero and after delivery? Which one is higher? Eh? Oxygen, partial pressure of oxygen is very high in the womb, higher than that of outside. Why is it not higher? Eh? Plenty of oxygen inside the womb. Because there is oxygen. Oxygen is found where? In the air. So that's why God has made hemoglobin F to special, irrespective of the amount of oxygen in the placenta, the hemoglobin F is adapted to go and pick it, then take it to the baby. You understand? So that is why it is higher. And after birth, there will be no need for that function of hemoglobin F again. Would there be need because there is oxygen in the air? Would there be need for that function again? No. So that is why there is a switch from hemoglobin F to A1 after birth. Okay, so when does it switch? When does it take place? Do you have idea? The switch after delivery, that you now have 20% of hemoglobin A instead of 80% of hemoglobin F. What months? In 6 months. It is actually 6 months. So by 6 months, the value declines gradually from 80% to like 70, 60 of the delivery of the baby to like 20%. I'm talking about hemoglobin F now. By 6 months, because it's not, the function is not needed again. There is plenty of oxygen in the air. Okay, so and that is why you have that. So this is like I was trying to say. This is chromosomal level. You have the alpha. You can see chromosome 16. It has zeta 2. So that's the alpha, and then the alpha 1 and alpha 2 is located on chromosome 16. How many amino acids do you have on alpha and alpha like? 141. 141. How I know it, I use a formula for it. 16 is higher than 11. So if the one that has the higher number has the lower number of amino acids. Okay, so 146 is 11 chromosome, 146 for beta, and so all the beta like and the beta are on chromosome 11. You can see beta, delta. You see there are two types of gamma depending on whether it's alanine or glycine at position 136. So gamma can be gamma glycine or gamma alanine. Okay, then you have the zeta 2. No, you have the epsilon. It's not clear, epsilon. So you can also have a combination during embryonic as early as 6 weeks of pregnancy. Like I said, I was going to genotype a 6 weeks pregnancy to determine the genotype. So this is what is operational. These are the hemoglobin in embryonic stage. It's called GOA1, GOA2, and Portland. If you look at it again, it's a combination of either alpha or alpha-like and beta or beta-like. It cannot be two alpha, I mean, it cannot be alpha and alpha or alpha-like. You understand what I'm trying to say? It cannot be alpha and zeta. Okay, it cannot be alpha and zeta. Like the GOA1 is zeta 2, it always appears in pairs. Zeta 2 and epsilon 2. GOA2 is alpha 2 and epsilon 2. Okay, so it's either alpha or alpha like and beta or beta like. Then the Portland is zeta 2, gamma 2. So these are the embryonic hemoglobin. So after this phase, then they are gradually replaced by hemoglobin F. After this phase, they are gradually replaced by hemoglobin F. And by 6 months of delivery, there is a switch again from majority hemoglobin F to hemoglobin A1. A1, majority. What's the percentage of A2 in you and I if you are A? Between 1.5 and 3.2. The hemoglobin F level drops to less than 1% because we don't need it again. Okay? So this is the HB Portland. That's the embryonic zeta 2 and gamma 2. You can see zeta is the alpha like there and gamma is the beta like. Okay? Then HB GOA1 is zeta 2 and epsilon 2 and HB GOA2. Like I said, there are two types of gamma chains depending on whether they have alanine or glycine at position 136 rather. Okay, so this is another genetic diagram of the alpha like and the beta like. Okay, you can see all the alpha like. This is the zeta, this is psi zeta. They are called pseudogenes. I'll talk about it later. This is psi alpha, this is psi alpha, this is alpha 2, alpha 1. That's theta, theta. That's theta. It's also alpha like. Okay, and the beta, they're also epsilon, gamma, whether it's glycine or alanine, psi, beta, delta, and beta. So located on chromosome chromosome 11. Okay, so this is the site of hematopoiesis. There are typical MCQ questions. Whether where is hematopoiesis? Where does it take place? Between 0 to 2 months, between 2 months to 7 months, and then between 5 to. I want to ask, have I taken your iron metabolism in the previous? Okay, okay, I forgot. So in infants, the bone marrow is used for, the long bones are used for hemopoiesis, and during hemopoiesis, hemoglobin, erythropoiesis, and then hemoglobins are formed. Okay, but in adults they are the central skeleton. So that's why we do. Have you watched us do bone marrow aspiration before? We want to make a diagnosis. No. So we don't know the site we use for the bone marrow aspiration. You don't know the site we use for the bone marrow aspiration. We use the central skeleton. So, like, we use the anterior superior iliac spine. We have a special needle, not the needle for injection, thicker and bigger. We'll put it inside the anterior superior, like 2-3 cm away from anterior superior iliac spine, and then aspirate the contents of the marrow and make a smear and stain. We are trained as hematologists to give diagnosis. Okay, but if you don't want to use anterior superior iliac spine, we use posterior superior iliac spine. Okay, and at times, if you are bold enough, we use the sternum, and if you put your needle and goes into the heart, you are on your own. So we use proximal end of the femur. It can be anterior superior, that's the pelvic. But in babies too, babies will also do, because the bones are very fragile, we use the tibia bone occasionally for babies. We just put the needle, a small needle for babies, put it and then aspirate. And most of the time we do it under local anesthesia, because it's not easy to insert a needle into somebody's bone. No, ordinary injection. So this is what happens. This graph, you can see the alpha chain. This is before birth. Look at this. Everybody look at it again. This is 6 weeks. 6 weeks of gestation, of pregnancy. So the alpha chain goes up and then plateau until death. Okay, but look at the gamma chain. Gamma chain goes up until like 12 weeks, then gradually comes down, and then comes down until 6 weeks, or 6 months rather, 6 months, it then goes off completely. Okay, so this is the zeta. You can see zeta drops. It's an embryonic hemoglobin. So by 10 weeks of pregnancy, it has disappeared, okay, completely. So don't forget, then the beta rises gradually until delivery, and when it's like 12 weeks, and then like, so that's why you could have. So what's the combination in hemoglobin A1? I want you to remind me again. Alpha 2, alpha 2, beta 2, hemoglobin A2. Alpha 2 delta 2 is A2, alpha 2 delta 2. What about hemoglobin F? Alpha 2, gamma 2. So depending on the age at which you are considering, and that is why you have this combination due to different ages. Okay, so these are the embryo. I know you will not remember this, I just so I didn't bother to ask you. The HB Goa, HB Portland, Goa 1 and Goa 2. Goa 1 is zeta 2 epsilon 2. I hope you remember the MCQ questions. Goa 2 is alpha 2 epsilon 2, and HB Portland could be zeta 2 and gamma 2. And like I said, the gamma can be either glycine or alanine at position 136. Okay, I've explained this. I said, okay, you want to have a mitosis, you have to have another cell. So during interphase, that's where this takes place. Before you have another cell division, the cell must be ready to produce another DNA that will be used by the new cell. So it is during interphase, during cell cycle, that you have replication, and that's what is happening. So the replication is inside and we don't have to talk about details of replication here, because this is not a biochemistry class and you have done that. So but you should know that you have your DNA replicated normally, okay, and then it is transcribed. And why do you need to transcribe? Because the DNA, the size of the DNA is too big to move out of the nucleus, because translation, protein synthesis takes place in the ribosome, and ribosome is where? Is in the cytoplasm. Ribosome is in the cytoplasm. So God has made it in such a way. So what's the idea behind the, replication and transcription? The idea is that it's coiled, double helix coiled. It is paired, and then you have one copy of the chain copied like in a complementary manner in RNA. That's why you have messenger RNA, produced from one strand of the DNA in a so the size becomes smaller, so it will be able to move out of the nucleus into the cytoplasm and finds its way into where? Ribosome for protein synthesis. Okay, so that's what is happening there. You have the replication to transcription. Transcription will now, but before it moves out, it has proofreading ability. It can correct all its errors, okay, and that's why this splicing, so that by the time it moves out, you have a well-formed mRNA, okay. So you have the what is called the introns that are non-coding and the exons that are coding. So the body will slice away all the non-coding parts in the mRNA before it moves out. Okay, you are going to see that picture. Okay, so after that, we're going to unite the coding, because it is the coding parts that are interpreted by your transfer RNA to bring the anticodon from the attached to the one search for amino acid and make synthesis of amino acids. So there is no need and no point keeping useless part of the mRNA, which is the non-coding part, and that's why the slicing. I'm not sure you understand what I'm talking about. Okay, you don't. Your biochemistry is defective. Okay, so we are going to see the picture better. So like I was saying, you have but replication takes place in the nucleus. Okay, you agree with me? Okay, and then transcription takes place in the nucleus. Okay, so after the transcription, which is a copy, complementary copy of each strand of the DNA, okay, So it becomes smaller. The messenger RNA becomes smaller, and it is processed in the DNA. The processing is to cut off the non-coding parts. I hope that is clear. In the nucleus, you want the body will cut off the non-coding part and then merge the coding part and become smaller again, so it is, you know, sent out of the nucleus into the ribosome, and in the ribosome protein synthesis of the polypeptide chain, the globin, the alpha, the beta, the delta, the zeta will start, you know, based on the nucleotide arrangement in the DNA. I hope it's clear. Based on the nuclear, and I said this morning that if you have a protein, it doesn't fall from heaven. There must have been a messenger RNA, and there must have been a DNA. Okay, so if you have a DNA, the nucleotide arrangement in the DNA, so if there is no point mutation in which you have thymine replacing adenine, so what type of hemoglobin are you going to produce? No thymine, normal hemoglobin. But if you have thymine replacing adenine, you are going to produce hemoglobin S, because your end product of the protein, you have valine replacing glutamic acid. I hope that is clear. So it is regulation takes place in the nucleus of the regulation of the replication and transcription. So this then is followed by a scission of the introns. This is what I'm trying to say. You have the DNA, and after this one, you now have your mitosis. The cells will divide, and the produced DNA and protein will now go into the new cells. That's what happens. Okay, so you have two cells in mitosis. So you have transcription, and then you have your mRNA, messenger RNA after the transcription. And don't forget, the non-coding parts will be spliced off, and then you have merger of the coding part. So it becomes smaller, and this moves out into the where? Cytoplasm. Where in the cytoplasm? Ribosome. Why? For what? For protein synthesis. Okay? And this is what happens. This is just the genetic structure again. You don't need all this. So this is what I'm talking about. The DNA will now be in this initial transcript with mRNA. You can see two color. Okay, so before it is sent out to the cytoplasm, all the introns, which are the non-coding parts, are spliced off, so that you make it even smaller, and the body does not send what it does not need into the cytoplasm. So you now have mRNA containing only the coding parts. Okay, is this difficult to understand? So this is the transfer RNA in the this is the code, this is the codon in the mRNA. This is your transfer RNA that has the anticodon that must be complementary. So it brings amino acid that has anticodon to this codon, and then the synthesis starts gradually. So that's what is called transfer. Bringing transfer RNA brings is an RNA that brings amino acid that has anticodon to specific codon arrangement on the mRNA that is produced from the nucleus. Is that difficult to understand? So okay, and that's where synthesis. That is how alpha chains, beta chains, delta chains, gamma chains, zeta chains, epsilon chain, all the chains, all the alpha, and that is how they are produced. That is how they are produced. So this is the synthesis, protein synthesis. You can see the transfer RNA is bringing the anticodon containing the protein, containing the amino acids. So if you have protein energy malnutrition, kwashiorkor. Will you have adequate amino acid for hemoglobin? No, you won't have. That's why you must eat good food, balanced diet containing protein, because it doesn't fall from heaven. It is what you eat. You are what you eat. Okay, if you, so you can see babies that lack, or mothers that lack good food, so the PCV will be very high. Is that correct? It will be no, there will be no, it will be low, because they cannot produce polypeptides. And that is, we are going to talk about role of iron too. So, and that is why you need all this information, and then you'll be able to appreciate the. So we'll talk about him. There is no e after the m, so it's an error. There is no him. It's called him. So him, like I said, how many him do we have in hemoglobin? Okay, so you have 4 heme, but this is just a structure of 1 heme. You can see the structure has something in the center. Iron, it has iron. Iron must be in ferrous state. I've just, how many Fe2. So, but like I said, some drugs can convert ferrous to ferric, and then the heme will be very useful when it's converted. Is that correct? Okay, so there will be anemia too. There will be cyanosis. Okay, so this is the structure of the heme. Don't let's not forget the polypeptide, the alpha chain. So after use, after the hemoglobin is destroyed, the polypeptidase will now break down those polypeptide again into various amino acids, and then it can be. So what's the lifespan of red cell? So where are they destroyed? Spleen, liver, and the macrophages. Okay, so they are destroyed, and then the hemoglobin is released. Okay, once the hemoglobin is released, the alpha chain, the beta chain, all the chain will now be broken down to the various amino acids for reuse. And then the heme will also be broken down, and then you have iron separately. Pyro ring, so that's 4 pyro ring. These are the pyro ring, pyro ring, and in the center it has iron. So if you are iron deficient, will you have adequate heme? Because you have heme, you have pyro ring, and there is no iron, so you can't bind oxygen, and that's why pregnant women take iron to aid production. If you take iron and you are not taking adequate protein, will you have adequate polypeptide chain? No. I hope we are on the same page. Okay, this is the heme. Again, this is the structure. It has 4 pyro ring, A, B, C, D, and it has iron inside it. So this is the heme. This is the structure of the heme. Okay, and this is where the polypeptide chain binds to. It has a another ring that binds the polypeptide, the chain, the alpha chain, the beta chain, and don't forget it is folded. The heme is folded inside the poly. You still remember the primary structure of protein,